Thank you to the organizers of BioAnt Talks for hosting this virtual symposium, and thank you all for your attendance. My name is Janna Andronowski, and I'm an assistant professor of biomedical sciences at Memorial University and the forensic anthropologist for the province of Newfoundland and Labrador. Today, we're going to examine the past, the present, and the future of the analysis of bone microstructure and its importance to bone biology and biological anthropology, specifically forensic anthropology and human identification. I will be providing an overview of my research efforts in virtual histology with a focus on the third and fourth dimensional realms and highlight the potential to integrate emerging new technology into the study of bone. I want to note that certain work presented here is supported by the National Institute of Justice and that the opinions, findings, and conclusions expressed here are my own and do not necessarily reflect those of the Department of Justice. I'm going to begin with the big picture and provide an overview of modalities used for examination of human skeletal remains in forensic anthropology, specifically as they relate to estimating age at death. And traditionally, this employs a macroscopic or morphological evaluation. But another approach employs a microscopic assessment. And at the crux of our histological age estimation methods is the process of bone remodeling. I will briefly discuss how this process of bone turnover provides the basis for our methods. The presentation will further highlight various case studies that I'm currently involved with or have been working on at the Canadian Light Source National Synchrotron Facility to highlight the diversity of applications we can achieve using this technology. Lastly, I will discuss ongoing work that I'm pursuing in my current academic appointment involving in vivo time-dependent 3D and 4D imaging using live animal models. In forensic anthropology, our macroscopic methods focus typically on the assessment of gross skeletal features. So for example, the pubic symphysis, auricular surface, or sternal rib ends. But in the forensic context, as we know, human remains are often frequently highly fragmented, and this can make our conventional macroscopic age determination methods ineffective. We can turn to medical imaging, for example, to identify age at death, particularly for juvenile unknown skeletal remains. Looking at tooth development and eruption is more closely associated with chronological age than other parts of the skeleton, since we are familiar with regular formation and eruption times. In addition, several histological methods have been developed that use cortical bones specifically, as pictured here in the lower right-hand corner, to determine age at death based on the evaluation of microstructural features that are the products of bone remodeling. My work is a combination of the latter two, medical imaging and histology. And I want to note that a common limitation of our current two-dimensional histological techniques is that they're inherently limited by their two-dimensional nature. This provided a limited window into the inner workings of bone because bone exists in 3 dimensions and remodels over time, introducing a 4th dimension. We've seen advances in high-resolution imaging that are enabling the shift from 2-dimensional to 3-dimensional analyses of bone microstructure. And this in turn is providing new insights into the remodeling process. So for example, in the image here on the bottom left, we can see a 2-dimensional cross-section of human rib bone, and we can appreciate the basic structural units or osteonal systems and the reversion canals that carry vasculature in their center. But if we take a step further and look at this in high resolution 3D, not only can we appreciate the basic structural units, but we can see how they interconnect with surrounding units, how those reversion canal systems are also interconnected. So these 3D reconstructions of reversion networks will really enhance our models that seek to describe the regulation of cortical bone turnover. 2 primary research questions that are ongoing in my lab are, can we account for the range of stimuli hypothesized to alter bone microstructure in histological methods? So a lot of our methods do not account for physiological bone responses to things like metabolism, the foods we eat, the substances we ingest, including alcohol and drugs, for example. So how can we improve our methods? I'm looking at using 3D imaging technologies as well as live animal models to answer new questions in forensic anthropology as well as in human health. And so before we get there, I'm going to provide a really brief overview of the bone remodeling process that forms the basis of our 2D methods to then introduce our various 3D imaging modalities and where we're going from here. Bone remodeling is the mechanism by which mature bone is replaced with new bone by the coordinated action of our osteoclasts that resorb bone and our osteoblasts that form bone. And this occurs all throughout life at a predictable rate and occurs in all bone types. What you're seeing pictured here is a basic multicellular unit or BMU of bone. And we can see that there's a resorption tunnel or cutting cone that is osteoclast at the leading end. Okay? And this cross section represents the 2D histological view you're seeing on the left. So this resorptive area would be represented here. The filling cone that travels behind contains our osteoblast cells that deposit bone in concentric lamellar rings, giving rise to new basic structural units or new osteonal systems. And remodeling is critical because it helps to adjust our bone microarchitecture to meet changing mechanical needs. It helps to repair microdamage in the bone matrix and prevents the accumulation of old bone that can lead to micro and then macro fractures. And we're going to talk a lot about synchrotron microcomputed tomography today. And I wanted to include this SR micro CT longitudinal view of the same set of structures, the BMU. And this allows us to see really fine details as that BMU tunnels through bone. So here is our closing cone behind, the leading resorptive area, cutting cone in the front with those leading osteoclasts. The premise behind our histological age estimation methods is based on this evidence that bone turnover is occurring at a predictable rate over an individual's lifespan. And the length of time in which remodeling occurs will be the primary influence on how many osteons that we're seeing accumulating per unit area. So this includes both fragments and intact osteons. So here in the image on the right, we can see A is an intact osteon and B would be representative of a fragment because that Herbergen system or canal is obscured. The key to understanding our methods is to really make sure you're accurately defining an intact from a fragmentary osteon, for example. And we apply these methods to both our femur and rib, typically in forensic anthropology. These are the samples we have based on former availability of biopsy specimens, for example, in the 60s. One of the variables that we quantify is called osteon population density. So this is the count of remodeling events that we see divided by the cortical area of bone. But these osteon counts are limited in their application to older individuals because we do see that an asymptotic value is reached where our intact osteons and fragments crowd the cortex. Cortical area measurements are also employed. But again, this can be a subjective measurement because the observer is having to determine where cortical bone is ending and trabecular bone is beginning. So although we've learned a lot from our two-dimensional histological methods, there are issues that demonstrate, particularly with reliability and accuracy in terms of sort of avoiding specific factors like extrinsic and intrinsic biological factors and their varying effects on bone microstructure. So for example, we don't account for physical or chemical destruction. So diagenetic change within a bone cross-section, it's simply skipped over, as well as the influences of hormones such as estrogen, parathyroid hormone, and different disease processes like osteoporosis, as well as the effects of alcohol and drugs. And this is sort of the focus of my research trajectory at the moment. We further have issues with existing methods in terms of how variables are defined, as well as where on the particular sample, a histological age estimate is read. There's also the chronic problem of underaging young adults and overaging older adults. Age reflects time. Time is the ultimate linear variable, but we're forcing age-related processes into this linear model. I like to use this graphic from Crowder et al. 2017 that displays a representation of, on the left, a young individual bone sample. If you were looking through the reticle of your microscope, you'll notice that these circles here, the osteons, are larger and there are fewer of them. Whereas in our older individual, we can see more numerous osteons, they're smaller. But if we're applying, in this case, the Thompson method from 1979, we'll be counting the hit points, so these black dots within the given cross-section. And in both of these examples, there's a count of 14 hits. So this would result in identical age estimates, although there's a hypothetical 20-year age difference between known individuals. So what can we do? Where can we go from here? As I mentioned, I'm currently trying to investigate how we can account for a range of stimuli that we know affect the bone remodeling process. But can we do this? Not currently, right? So we need to really consider the dynamic nature of bone, and it's a great avenue for new research, as well as for potential student involvement. So let's take a look at what new questions we can answer using 3D imaging technologies. Early micro-CT work provided basic information about the 3D dynamic structure of cortical bone tissue. We can deduce a lot of information from that. So for example, we can see a full 3D reconstruction of the rib here and extract things like trabecular architecture, isolate the pore network like in the bottom left, as well as look at distance between pores, distance map, for example. But this is taken with a desktop micro CT system. And these systems don't allow for the detection of a lot of other detailed features, such as bone's cellular network. So we can do better. We can go a step further. We can use synchrotron micro CT technology, and a synchrotron generally is a specialized form of circular particle accelerator that uses a source of brilliant light. And this enables scientists to study the microstructure and chemical properties of materials, and also offers new insight into the studying of different material properties, such as bone. For example, we can investigate bone quality, bone quantity, microstructural features, and it's proven very effective for imaging the cellular network on a very small scale, for example. I've included sort of a spattering of my favorite synchrotron facilities. We have ESRF on top here, that's in France, the Canadian Light Source, which is where I do a lot of my research, as well as Spring 8, which is located in Japan. The Canadian Light Source is located in Saskatchewan, Canada, and you can see it all lit up here for its 10th anniversary, but it wouldn't be Saskatchewan or Canada without showing how it looks a lot of the year, negative 40 and cold, but we can do a lot of really amazing science here. This is taking a look inside the storage ring. And so we can see here the storage ring from the mezzanine level. This is where the electrons will circle around close to the speed of light and are directed down laboratory beam lines to end stations where the researchers will then control for the wavelength of light, as well as our particular experimental parameters. And the ability to probe these sort of histological structures non-invasively at this very high level has really increased our anthropological applications. So for example, there's been projects looking at comparative anatomy, experimental design, discovery-based approaches that can really illuminate, bone growth, physical adaptation, health for applications across not only biomedical sciences, but bioarchaeology and forensic science, as well as anatomy and paleontology. So here's a quick shot of the biomedical imaging and therapy beamline, or BEAMIT. It is designed specifically for imaging biological tissues, and it's only one of 4 worldwide. And this is where I do the primary experiments for my research. We can employ setups such as X-ray imaging tomography, as well as X-ray fluorescence imaging. So I'm going to be providing some examples here in this first area, micro CT, and then briefly discussing some anthropological relevant studies that we are pursuing at the Canadian Light Source. So in terms of image acquisition, I wanted to first generally describe how these experiments are run at the BEAMIT facility, and then discuss 2 recent projects that I've worked on that employ this setup. So here we can see inside the hutch, there's a custom built stage and a white beam microscope setup that's designed for imaging various materials, biological tissues from animals and plants, metals, ceramics, and it can be adapted for various experimental setups. We'll mount each of our specimens on a goniometer, as seen here, and it will be centered in the lab so it stays within the field of view during imaging. This is the X-ray tube here. Images will be obtained using that white beam microscope setup, as I mentioned, with a pixel size of 1.5 micron, and we take approximately 3,000 projections spanning 180 degrees of rotation. So we can watch our sample from the control room. So here's an example of that goniometer on the custom built stage, and the arrow is pointing to our bone sample there. Once our scan is done, we can take a look at the data, and this is an example of a single projection from a human 6th rib sample. We can see a cortical bone here and trabecular struts here. These represent the haversian or vascular canals. And then all these sort of tinier spots are our osteocyte lacunar spaces, where the osteocytes reside during life. So we take a sort of region of interest, we can really appreciate, again, those haversian canals. We can see even the detail of the cement lines delineating basic structural unit and the osteocyte lacunar spaces here. Our vascular canals and our osteocyte lacunar spaces are then separated from the high density bone using various image processing workflows. So this is a very general examples showing sort of despeckling that's conducted to remove noise, and you're seeing an X, Y, and Z sort of plane representation once that segmentation has been applied to both our vascular canals and the osteocyte lacunar spaces. And then we have a rough three-dimensional render, again, canals in blue, osteocyte lacunar spaces in red. We can create three-dimensional videos that, again, highlight those air spaces within the bone for visualization prior to doing our data extraction. And here we can see two more examples of a younger female, 21 on the left, 88-year-old female on the right. And again, you can appreciate the increase in size of the vascular canals, the increase in connectivity in the older female, as well as in that two-dimensional cross-section on the top here. So now that we've discussed sort of that brief introduction to how imaging is conducted, I wanted to talk about a few case studies that are relevant to forensic anthropology. And the first one is using 3D imaging to assess bone cellular network and how this might glean information about nuclear DNA yields from bone. So forensic scientists typically sample dense cortical bone from the shaft of the femur or tibia in a forensic identification context. There's been various studies that have focused on examining DNA yield by element. A lot of these are retrospective in scope, and the researchers didn't evaluate all element types within a single skeleton. And particularly our smaller, predominantly cancellous bones are excluded from these analyses. So in 2014, researchers revealed that there may be variation in the amount of recoverable nuclear DNA among skeletal elements, and looked at different tissue types, cortical versus cancellous, in a specific skeletal sample from the University of Tennessee in Knoxville. Their research revealed that there was variation and there was higher bone quality and quantity of nuclear DNA in these untraditionally sampled smaller cancellous bones, like the patella, for example, phalanges of the hands and feet. But the researchers were not able to pinpoint sort of a reason for that. So I collaborated with Dr. Amy Mundorf, Donnie Stedman at University of Tennessee to turn to bone microstructure to help explain potentially what we were seeing. So I completed a survey of osteocyte lacunar variation because our osteocyte lacunae are these divots within bone. They serve as proxies during our X-ray process for the actual cells themselves, since we are looking at dry bone in our experiments. And I used the exact same skeletal sample as the original authors and investigated whether we had differences in the lacunar density or the cellular density in these different bone types. So results demonstrated that osteocyte lacunar density was actually independent of nuclear DNA yield, suggesting that there was an alternative explanation for what we were seeing in these bones with higher cancellous content. So we take a look at the images here. On the top left, this is cortical bone. We can see our vasculature in red, our osteocyte lacunar spaces in gray here. Cancellous bone looks a little bit different. So we can see the trabecular struts here. Osteocyte lacunae are in blue. Again, they're much less numerous quantitatively, is what we found. However, we take a look at a two-dimensional cross-section of the trabecular bone region of interest. We can see the trabecular struts here, and all this sort of mottled gray tissue is soft tissues that remains within the dry bone. So if we take another look at the full three-dimensional render of this, we can really appreciate all of that soft tissue. Again, here's the bone in the darker gray, and all of this gray mottled material is the soft tissue. And if we zoom in on that, you can see that a little bit more clearly here on the right. So I went a step further and looked at X-ray photoelectron spectroscopy to confirm the presence of elements that are common in soft tissue. So for example, carbon in these specimens. And this was confirmed that these are likely soft tissues. And this is what was likely driving up our nuclear DNA yield in this specific skeletal sample. A second case study features the analysis of bone microstructure in the study of the occurrence of osteon banding among human bone types. And I demonstrated in a previous study that we can use synchrotron micro-CT as a valuable tool for 3D reconstructions of osteon banding. So these osteon bands are commonly described as an indicator of non-human bone in the anthropological literature, and that they're not commonly associated with human tissue. However, my CLS experimental work, I did find that osteon bands were observed regularly, especially in the flat bones of the cranium, so temporal, parietal, occipital, and even mandibular specimens. For example, And in these elements, the bands ranged from 5 to 12 osteons in length and were comprised mainly of primary osteons. In the long bones, so femora, tibia, ulnae, isolated bands were documented as well as instances where osteons displayed linear patterns that did not form distinct rows. So here's an example of a longitudinal section through a bone sample. This is from the temporal. And I assessed each image stack for the presence of osteon banding, as well as the number of osteons per band and the number of bands per specimen. Here, for example, we can see a larger resorptive area in the darker gray with a number of osteonal systems sort of stemming from that would then, depending on where the cross-section was taken, result in this linear-oriented set of osteons. So again, we can learn a lot more about the three-dimensional structure of bone instead of just that one picture in time, that one two-dimensional cross-section. And if we do a three-dimensional render of that same osteonal system, again, with the many osteonal segments stemming from it, we can see a high degree of interconnectivity between these in the vascular network, as well as how they're represented in a two-dimensional cross-section. So an encouraging area, for me at least, for future research is to understand the variability in the interconnectivity of the vascular canals that we're seeing in human bone and the branching patterns among different types of skeletal elements, flat bones, short bones, long bones. So we've answered a few questions now in terms of 3D bone imaging techniques in forensic anthropology. We've looked at some new dimensions, but what about the four-dimensional realm? So this is 3D change over time. So the preliminary in vivo work that I've been doing in my lab is at the stage for the next phase of my research program that's looking at the effects of prolonged drug use on the remodeling of cortical bone in an animal model. So we've talked a little bit about how limited data exists relating to the physiological processes that we know affect bone. And this is true for the longitudinal impact of opioid abuse or substance use on bone remodeling. So we need to further understand the underlying biological processes in order to improve the applicability of the histological age estimation methods that we use in forensic anthropology, as well as the scientific standards within the field. So this current work is part of a large-scale NIJ-funded project that employed both a novel microcomputed tomography approach and a dynamic histomorphology approach to evaluate opioid animal model of longitudinal bone remodeling. And the results from this micro-CT portion was presented during the AFS annual scientific meeting in 2021, combined with the dynamic histomorphometry that was presented in 2022 at AFS. And I will briefly summarize our findings here, as well as the model. So before I can sort of discuss the proposed work, I wanted to talk about sort of a primary aim here of the project, and that is answering the question of how do analgesic drugs, in this case morphine sulfate, fentanyl, affect microscopic structures of cortical bone using histological age estimation methods in forensic anthropology. And we can do this again using a live animal model because we can control for confounding factors, for example, such as biomechanical stressors, diet, housing, these types of things. So as you can see in our timeline, we are using New Zealand white rabbits because they have a spontaneous bone remodeling process comparable to humans. They have secondary osteonal bone. We had 3 experimental groups, so a morphine sulfate group, a transdermal fentanyl group, as well as a control group. And we administered either drug to our experimental groups for an 8-week period. And following euthanasia, we extracted the long bones from the lower limb, the femora and tibiae, and employed desktop microcomputed tomography, as well as dynamic histomorphometry. Our hypothesis was that opioids would significantly alter microstructural features using histological age estimation methods in bone. I wanted to show you a brief summary of the preliminary findings. So these are 3-dimensional renders of femora and a longitudinal 3-dimensional render of a femur from our control group. We can see in this strong patterning of pore networks compared to our drug groups. And I'll talk a little bit about how they differ, but we can see some concentrated areas of remodeling. For example, here on this aspect of the bone, the color scale, reds, yellows, greens represent larger pores, whereas blues and darker blues represent smaller pores. In our morphine animals, we can see less sort of consistent patterned cortical bone distribution. So for example, our pores, although smaller in this case, are more numerous. And we also saw a higher percentage of closed pores higher surface area ratio to pore or cortex volume, and just more individual pores. In our fentanyl animals, again, we're seeing a far increased number of these smaller pores, again, in these lighter blue, as well as a smaller degree of connectivity, so less linear organization of our vascular canals. So not sort of the larger resorbed events that we might have hypothesized, but more overall porosity. So in general, opioid trends, as I mentioned, we're seeing an increase in pore frequency in our fentanyl and morphine animals. So for example, more individual pores, higher percentage of closed pores, and the pore size, they're smaller. So the mean thickness and separation was also smaller, but there were more of them, as well as we saw less linear organization in the connectivity, as well as increased connectivity density. But there was also this less consistent, more dysregulated pattern of cortical bone distribution. So what does this mean for forensic practitioners? So again, we know that these life history variables, drug use, in this case, opioids, alcohol, diet, nutrition, does affect the bone remodeling process and therefore age estimates. So we recognize that low osteon counts can be associated with substance abuse from previous studies. So for example, Karen in 2009 looked at the effects of methamphetamine on bone turnover and found that, again, these individuals are being underaged. So what I'm trying to answer then is this prolonged opioid use that we're seeing discernible in the cortical microstructure used in our histological age estimation methods. And so far, the results indicate that yes, it is impacting our osteonal systems, the porosity that we're seeing in the bone, and overall fragility and quality. So going back to my main research question, then, can we account for the range of stimuli hypothesized to alter bone microstructure in our methods to be continued? So I'm continuing to work on this. Applying our in vivo animal model, for example, is really the next step. So in summary, we've learned a lot from our traditional two-dimensional histological techniques, but they do provide an inherently limited window into the inner workings of bone. Moving into 3D visualization and analysis holds the potential to improve our understanding not only of bone biology, but refine forensic anthropological applications and methods. But the future really is in this four-dimensional tracking of bone remodeling events that has the potential to be transformative for our understanding of bone physiology, controlling for confounding effects, and it really is the future. So with that, I'd like to thank my fantastic trainees at Memorial University, as well as collaborators at the University of Saskatchewan and Canadian Light Source Synchrotron. And I'd like to acknowledge my funding sources, specifically the National Institute of Justice and a recent grant from the Canadian Institutes of Health Research here in Canada that will further the opioid research that I'm working on in the lab. And if anybody has any questions, feel free to contact me either by email or on Twitter, and I look forward to hearing from you. Thank you.